Welcome, everyone, to another edition of our Frontage Laboratories podcast. I'm your host, Brian Newman. Thank you very much to all of our listeners tuning in today. Our topic that we will be covering is in vitro BE studies in ANDA drug product development. I am joined today by our special guest, Vice President of Analytical Services here at Frontage Laboratories, Kang Wang. So to start, Kang, can you please introduce yourself and explain your interest in vitro BE studies and ANDA drug product development?
Hi, my name is Kang Wang. I'm the Vice President for Analytical Service in Frontage. This is my 14th year here in Frontage. My group supports pharmaceutical drug product testing in different phases, from early IND development to final commercial drug product testing. My group also supports quite a lot of ANDA drug product development projects, including reverse engineering, dissolution, diffusion study, different formulation, comparison among tested products and the reference product, stability on development batch product, etc. We all know the criteria for ANDA product is bioequivalent between reference listed product, or called RLD, or branded product and a tested product, or generic product. Most of BE studies are performed in clinical center with a lot of human subjects involved, but in vitro BE can be done in our lab. That's why we have extreme interest in this topic. As for myself, I am an analytical scientist by training. I have been working in pharmaceutical analysis for 23 years.
Thank you very much, Kang, for introducing yourself. We're happy to have you here today. Let's start off by talking about my first question for you. What is an in vitro BE study, and what is the difference between in vitro and in vivo BE studies?
Before I address in vitro or in vivo BE studies, let's first talk about BE bioequivalence. Bioequivalence is a term in pharmacokinetics used to assess the expected in vivo biological equivalence of two proprietary preparations of a drug. If two products are said to be bioequivalent, it means they would be expected to be, for all intents and purposes, the same. When ANDA product is submitted to approval, BE between developer's drug or RLD and the developing drug or tested product is the central piece of information. Based on this definition, looks like only in vivo BE is required for submission. After we understand BE, let's talk about in vitro and in vivo. In vivo is from Latin, meaning within the living. It refers to work that's performed in a whole living organism. In vitro is also from Latin, meaning within the glass. When something is performed in vitro, it happens outside of living organism. From these two definitions, we understand the difference between in vivo and in vitro.
My next question for you is, what is the importance of an in vitro BE study, and are there any advantages of an in vitro BE study?
We already knew the difference between in vivo and in vitro studies. Clearly, in vivo study is much more complicated than in vitro. Recruiting appropriate human volunteers is critical for in vivo BE studies. It definitely costs more, not much longer, plus the results from in vivo are very variable. Sometimes it is difficult to repeat. In contrary to this, in vitro study can save cost, save time, and easy to control.
Thanks, Kang. On to my third question. Can you give me some examples of an in vitro BE study to our listeners?
From definition of BE, in vivo study seems the choice, but from above seen, in vitro has advantages over in vivo study. Does FDA accept in vitro BE study result in this case? The answer is no and yes. For most dosage forms, bioequivalence can be achieved using pharmacokinetic studies that measure the rate of absorption or bioavailability. You need to do in vivo BE study for those products. But for some special dosage forms, such as some semi-solid or topical products, or some oral dosage forms, their therapeutic function is localized or through binding to certain protein and molecules. Such products are not intended to be absorbed systematically by body. No pharmacokinetics study can be performed. For those products, yes, in vitro study is recommended by FDA or required by FDA. Based on guidance from FDA, topical products such as cyclovir ointment cream, lidocaine cream, and some solid dosage products like sucralfate suspension, cholestyramine oral powder, etc., do need in vitro BE studies.
That's really great information, Kang. Can you share some challenges to perform in vitro BE studies?
Challenges are coming from two aspects. First is experimental part. This whole in vitro BE study is lengthy and very labor intensive. It requires good planning and controls, such as concentration preparation, incubation time and temperature, sampling time, etc. Sometimes the concentration is low and the sensitivity becomes an issue. Different analytical techniques other than HPLC UV should be used. Second part is regulatory part. This in vitro study falls into GLP category. All the study protocol, study report, data analysis, such as statistical analysis, should follow GLP rules, which some regular pharmaceutical analytical lab most time is not familiar with these GLP rules. In contrast, most GLP lab, a lot of bioanalytical lab, they are not taking such a product testing project because they are not very familiar with product.
And my last question for you today is, what can Frontage do to help perform in vitro BE studies?
Frontage Lab has the advantage to perform such in vitro BE studies. Our company has long history to handle both GLP and GMP studies. Our QA and scientists are familiar with the regulatory requirements. For experimental parts, we have state-of-the-art analytical instrumentation. We are equipped with 84 French cells and can start several diffusion experiments for semi-solid in vitro BE studies. In the past, we have accomplished 2 in vitro BE studies for semi-solid, and the results were submitted to FDA. No deficiency was resulted from the submission. We are exploring in vitro BE studies for solid dose products with several clients. New study results from those solid products are expected to be produced in this in vitro category.
Thank you so much, Kang. That wraps up our topic today on in vitro BE studies in ANDA drug product development. I want to thank everyone so much for tuning into our Frontage Laboratories podcast. Don't forget to visit us at frontagelab.com to find out more information about our company and how we can turn our services into your solutions. Stay tuned for more podcasts in the near future. Stay healthy and safe, and have a great day.